mRNA technology, also known as messenger RNA technology, refers to a revolutionary approach in the field of molecular biology and biotechnology. It involves the use of mRNA molecules to instruct cells in the body to produce specific proteins. mRNA, messenger RNA, is a molecule that carries genetic instructions from the DNA in a cell's nucleus to the ribosomes, where protein synthesis takes place. The primary use of mRNA technology is in the development of vaccines, particularly mRNA vaccines. Traditional vaccines often use weakened or inactivated forms of pathogens, or specific proteins from the pathogens, to stimulate an immune response and provide immunity. However, mRNA vaccines work differently. mRNA vaccines utilize a small piece of the virus's genetic material, specifically the mRNA that encodes the viral protein of interest. This mRNA is encapsulated in lipid nanoparticles to protect it and help it enter cells. When the vaccine is administered, the mRNA enters the body's cells and instructs them to produce the viral protein. The immune system recognizes the foreign protein as a potential threat and mounts an immune response against it, which includes the production of antibodies. These antibodies can then neutralize the actual virus if the person is exposed to it later. The advantages of mRNA technology include its flexibility and speed of development. Traditional vaccines often require the cultivation of large quantities of the pathogen, which can be time-consuming and complex. In contrast, mRNA vaccines can be designed and produced quickly, as the mRNA sequence can be synthesized in the laboratory once the genetic information of the target protein is known. This makes mRNA technology particularly useful in the context of rapidly emerging infectious diseases, as was the case with the COVID-19 pandemic. It's worth noting that mRNA technology is also being explored for other medical applications beyond vaccines. Researchers are investigating the potential of mRNA therapies for various diseases, including cancer, genetic disorders, and autoimmune conditions. The idea is to use mRNA to deliver specific instructions to cells and trigger therapeutic effects, such as the production of proteins that can correct genetic mutations or modulate immune responses.